Good evening, everyone. We're going to get started in about another 2 or 3 minutes. I'm just going to give some time for other attendees to trickle in. And just let you know, the meeting is going to be recorded. So if anyone needs to hop off partway through because of kids or something's on fire on the stove, that's okay. Let's see if I can get.
So you're going to let us share screen because I couldn't send you this PowerPoint.
Yeah, so I'll transfer screen share power as I call people up for the presentation portion.
Perfect.
Okay, and again, for everyone just trickling in, we're going to give it another 1 or 2 minutes to let other folks come in, and then we'll get started, and this is being recorded. Okay, so I think the numbers are holding steady, or a very large number of people are having issues with logging into GoToMeeting. So we're going to get started. So thank you everyone for coming tonight. This is going to be an educational webinar talking about various research projects that are going on in the Great Bay system related to Pfas, also known as per and polyfluoroalkyl substances. Tonight I'll be moderating and I'll be giving a very brief overview of what Pfas are, and I'll do some of the housekeeping. But the main presenters tonight are going to be faculty from 2 universities in the state. So I'll introduce them in order that they'll be presenting. So the first is Dr. Paula Mauser from University of New Hampshire. So Paula, if you give us a wave. Followed by Dr. Celia Chen from Dartmouth College. And then Dr. Megan Romano from Dartmouth College as well. So the overview will be given by me. Paula will be talking about some of her research that is more focused on some fate and transport related to PFAS in the Bay. Celia will talk about some biological uptake into organisms and some of the research that her lab is planning on doing. And then Megan's going to talk about some epidemiological work she's been doing in her lab. Also tonight, we're going to have someone that's sort of off screen. That'll be Dr. Robert Thistle. He's a colleague of mine in the environmental health program. He'll be running technical assistance. So just some overview and housekeeping. This will be recorded. We will try to post this either on the NHDES website or onto our YouTube page in the coming days after the recording is done. There will be a Q&A session at the end of the presentations, but if at any time you just want to get your question out and get it written down before you forget about it, if you look in the little side menus, you should see one that has a drop-down tab for questions. Please feel free to write your question in there and send it on over to us, and we'll answer them in the order that we receive them. If you have any issues with either entering questions or any technical issues, please feel free to contact Rod at 603-271-1417. Again, that's 603-271-1417. Or you can email him at robert.thistle@des.nh.gov. So with that, I'm going to start giving just a brief overview of what PFAS are. I know a lot of folks in the audience may be familiar, but in case we have people that this is a new subject for them, just want to give them a primer. That way we're not burying everyone in an acronym soup. So what are PFAS? PFAS is an acronym that stands for per and polyfluoroalkyl substances. This is a family of man-made organic chemicals. It's estimated that there's over 4,000 to 9,000 different compounds in this family. That range of numbers changes based on how you define a PFAS. These formerly used to be called perfluorochemicals or PFCs, but that terminology's gone by the wayside. So some folks that have been doing this for a while may be familiar with that, or signs you'll see that if you do internet searches. Now, what sort of separates each one of these chemicals and gives them a unique property is their functional groups and the lengths of their carbon chains. So for example, we have perfluorocarboxylic acids in the family that have a carboxylic acid. So it's that little funny O and OH thing on the end. And as you either increase the zigzag in here or decrease it, so you make it shorter or longer, you get a new chemical. You get a new member of the Pfas family. But if we also swap out that funny OH for, say, the funny looking thing over there that has S and a bunch of O's on there, that's a sulfonic acid, we then get a different group of Pfas. So if you start getting creative, you can think of different ways that you create new compounds. Some of these are fully saturated in fluorines, where you see a lot of those Fs along there. Other ones are not fully saturated or partially saturated. Now, the reason that these fluorines are important to PFAS is it gives these chemicals a really unique property. It makes them very long-lived in the environment. They're really hard to break down and they resist heat and degradation. So sometimes we call these forever chemicals because they can last a very, very long time in the environment. But they also give properties that have been used in a lot of industrial and commercial goods. So one of those is aqueous film forming foam. That's a tool that's used to fight certain types of fires, specifically fuel fires. It's very effective at that because PFAS resists degradation under heat. So very useful, but again, if we use these like what was used at the former Pease Air Force Base or at other places that use this special foam, you can get groundwater contamination. But as we've done more and more investigation in the state, we've realized there's a lot of different sources of PFAS all over, from both industrial applications and manufacturing, but also from consumer products and things in our everyday life. So as we use these products in our homes, as we interact with these sometimes almost ubiquitous applications of PFAS, they get into our body, they can end up in our waste systems, or as these things get thrown away into the waste system, they can eventually cycle there, as PFAS will linger for a long time. So this is not an exhaustive list of all the places that PFAS can come from, but it's just an example where we're realizing these are in a lot of things we didn't think might be a source of these chemicals. Now, the reason we're worried about these is that they may be associated with certain health effects. So this is just a simplified overview of some effects that have been recognized by the CDC. That includes increases in cholesterol levels, liver enzyme levels that may be an indicator of liver toxicity, small changes in infant birth weight that are not ideal, some alterations in immune system function, increased risk of high blood pressure, preeclampsia for pregnant women, changes in both thyroid and reproductive hormone levels or their function, and potentially an increased risk for certain cancers. Now, this is something that's being studied nationwide, both by the CDC and its Agency for Toxic Substances and Disease Registry, that is a branch of that agency, but also by private and academic groups. And you actually hear about one of those academic groups tonight. So this is a constantly evolving area of research. You can find more information at the CDC's website, or if you want to contact any one of us, we'll try to get you in touch with an appropriate resource. Now, when it comes down to the Great Bay or any part of the environment, right, when we think about where these chemicals come from, they start in industry and they gradually move their way through these different systems. So this is a very simplified diagram of how PFAS move into the environment, but our ultimate concern is human exposure. And one of the things we're going to talk about tonight is how things can end up into the environment. And that specific environment we're going to talk about is sort of the Great Bay Estuary, but also our water resources in the state. So I'm a toxicologist for the state. I try to evaluate risk and how we can make recommendations to the state to address those risks. But what you're going to hear from tonight primarily are researchers that are helping provide that critical information that we need so we can make decisions or make the best assessments possible. So with that, that's going to cut my brief overview. Here's my contact information. It'll be available again near the end of the presentation. I'll be sure to send this out as a follow-up to everyone. If you have any questions, please feel free to reach out to me here, or again, you can throw them in the chat box. So with that, I am going to hand it over to Dr. Paula Mauser and let her give her presentation.
All right, can everybody see my screen? Let's see. How does that look? Good?
Yep.
Super. All right, good evening, everybody. I'm Paula Mauser, and I'm a faculty member at Civil and Environmental Engineering at the University of New Hampshire. And today I'm talking to you about municipal wastewater treatment systems and their discharge to the Great Bay Estuary and its relevance to PFAS in the environment. So I'm first going to give a little bit of a background on the characteristics of wastewater treatment facilities and relevance to PFAS. And then we're going to talk a little bit about what we're seeing in terms of research for what we're calling fractionation of these compounds within our wastewater media. And then I'll touch a little bit on implications for discharge to surface waters like the Great Bay Estuary and some of the ongoing research that we have that we hope to answer further questions. So as John mentioned, we have many different sources of PFAS in our environment, and municipal wastewater treatment facilities play this really critical role in what we think about as concentrating these compounds and then acting as a conduit back to the environment. And our wastewater treatment systems are so very integral to the removal of contaminants and pathogens before discharge back to the environment. If we didn't have them, we would have a lot more environmental contamination as well as disease in our space. And so in terms of PFAS, we have several different ways and sources that PFAS are coming to these key facilities. Of course, we have residential users, so municipal wastewater treatment facilities, they collect primarily from residential areas, larger developed and urban areas. But we also have discharge from industrial users, such as wastewater coming from landfills, so landfill leachate. We have septic haulers. We have certain manufacturing wastewater that comes to these facilities sometimes. And then within our residential and urban areas, we have other commercial users that might be discharging PFAS if they're doing things like metal finishing or certain commercial products that John mentioned. And so all of these different sources come into our wastewater treatment facilities, and then the facility is going to release media, hopefully cleaned media, either to the air, to an effluent that's usually discharging to a surface water body like the Great Bay or other waterways, and then solids, and we call those residuals. And those solids take another course. They can be landfilled or they can be processed in a way that allows them to be used for beneficial products such as biosolids for agricultural purposes. And so as you can see, there's a lot of different ways that the PFAS are entering this really important piece of our engineering infrastructure, and then hopefully we are collecting them and managing them in a way that they don't come back to the environment. I'm not going to go through the chemistry of PFAS, but I wanted to point out that there's something really important about the structure of these compounds that make them more amenable to transformations or biotransformations within our wastewater treatment facilities. And that is the type of functional group that they might have on them. So many of these compounds are fully fluorinated, or saturated, so all of those fluorines are connected to a carbon. But we also have polyfluorinated compounds, and those compounds are things like these fluorotelmer alcohols. And these compounds can have different functional groups on one end. And those particular ends are really more amenable to some sort of partial transformation. In addition, when I talk about some of our research, I'm also going to talk about long chain versus short chain, which differ depending upon the class and the size and the functional group. And And this is important because the composition of these PFAS make them want to attach to or more or less solids in our wastewater treatment facilities. So some of these like to go out in the solids form, and then others like to come out in the liquid form. And that's really important, and that's one of the focuses of this work. So, as you know, many of these compounds are very chemically, thermally, and biologically stable, which is why they've the nickname forever chemicals. But as I mentioned, some of them might break down partially to other compounds, and that makes them really difficult to track in our engineered systems and also in the environment. So here's a great example of why these partial biological transformations are important in our sewer sheds, which is where we have our wastewater being collected, in our treatment facilities, and in our wastewater residuals handling. We might have a fluorotelmer alcohol, something like an 8,2-FTOH. And our wastewater treatment facilities, one of the primary ways that organic waste is degraded is through what we call secondary treatment, biological treatment. And within this piece of our treatment facilities, there's these amazing groups of bacteria and larger organisms like yeast and fungi that are capable of enzymatically breaking down pieces of all different kinds of compounds. And they do this on purpose, and they might do it on accident just through other metabolic processes. So, this end of this particular fluorotelmer can be actually cleaved or transformed or changed in a way by biological entities. It generates an intermediate compound that we generally don't test for or measure. And then it can generate a couple of other compounds in a final step to a perfluorinated compounds. Some of these are compounds that are regulated, like PFOA. And so you can see how this could really complicate the tracking and the regulation of these compounds in these facilities when we have partial breakdown of what we call precursor compounds, like a fluorotelmer alcohol, to a terminal compound like PFOA, which potentially could be regulated in the future. So as an example of a review that came out a few years ago, if we start off with, say, 5 different raw Pfas compounds, fluorotelmer alcohols or acrylates, perfluorooctane sulfonates or alcohols or amines. So those are our precursor compounds. They can then transform to 8 different intermediates, which we may or may not measure. And then those also transform to another 6 terminal products. And so we go from starting with, say, 5 precursor compounds to generating 14 that we need to measure. So when we're talking about thousands of different compounds, this gets enormously complicated very fast. And it's really important because we want to know where these compounds are going in these facilities. So one of the questions we've been asking is, are there differences in PFAS concentrations and composition in our treatment facilities? And over the last couple of years, we have been looking at this in wastewater facilities that discharge to the Great Bay Estuary. So in 2019, March, one of my students, Ellie Tafazoli, went out and she collected samples at 6 different New Hampshire wastewater treatment facilities that are located in the tributaries of the Great Bay Estuary, and they're shown in the red dots here in this upper left map. We collected some information on PFAS in the influent of these wastewater facilities and the effluent, so where they're discharging to the tributaries in their area. And from that information, we went back and we resampled these facilities in July of that same year. And in our follow-up study, what we did is we looked at what was happening along the treatment train. So we took samples in the influent before biological treatment, which we call secondary treatment, after the biological treatment, after the disinfection process, and then before it discharges to the tributary. We also looked at the sludges that were generated, or the solids, during the wastewater treatment facility, and looked at PFAS in those solids. And we measured, we used what's called the isotope dilution method, or a modified method for wastewater samples, to target 24 compounds in these samples. But we also applied another assay that we call a top assay, and I'm going to touch on that a little bit, a total oxidizable precursor assay that helps us better close the mass balance loop when we have the potential for so many different compounds that can be transformed in these systems. And so, for those of you who don't know much about how your wastewater is cleaned, in the biological treatment processes of these wastewater basins, there's these great bacteria, and they live either in the presence or the absence of oxygen. And the facilities that we study focused on the utilization of both oxygen-rich and oxygen-depleted parts of the system so that we could look at all possible transformations that might occur or take place during the treatment process. So one of the facilities we focused on used something called an oxidation ditch, which switches back and forth between these two phases before it goes to a chlorination, dechlorination process. And then the other three facilities that we looked at used something called a 4-stage Barden-Foe, which is a more novel method that's used for also nitrogen removal at these facilities. And so alternating between low oxygen, high oxygen, low oxygen, high oxygen, and then chlorination and dechlorination. So in all four of the systems that we measured, we're looking at hopefully all possible reactions that might take place from the bacteria in these systems. And I mentioned, and this is really important for wastewater systems, that we are analyzing for these compounds in two ways. One way, that's a standard method, targets 24 Pfas that are generally the carboxylic acids and the sulfonic acids with some precursors. But we also applied to all of our samples something called this top assay. And the top assay is a way for us to basically pre-oxidize in the same way, chemically, in the same way that the biological organisms would, Pfas that might be transformable in our systems. So we take a sample, we chemically oxidize it with a really harsh hydroxyl radical that converts precursors to terminal products, and then we measure those. And we look and see if there's any precursors that we might be missing with our targeted analysis. Remember, we're only targeted between 18 and 24. So this helps us to understand if there's potentially compounds in there that we're not measuring that could potentially be generating some of these intermediate and terminal products. So we applied both of these analytical approaches. So let me give you a sense of some of the information that Ellie generated with this work. And I know this is a big slide, but one of the things that we like to do is we like to look across all of our samples to see if there's any general trends in the compounds that we're detecting in these facilities. And so along the left-hand side, going down, we see all the different Pfas that we measured, these 24 compounds that we measured in these samples, and they're classified as to whether they're precursors, sulfonic acids, or carboxylic acids. And then across the top row, we have the different facilities that we measured. And remember, we went out and we looked at influent, effluent in March, and then we went back again in July and measured influent and effluent. And so one of the things that you should see pretty quickly is that in most cases, we're detecting generally many of the same compounds across all of these different facilities that we're looking at. And that tells us that in a lot of our communities, we see, similar groupings of compounds coming into our municipal wastewater treatment systems, at least for the targeted compounds that we're looking at with our analytical techniques. The other thing that you'll see is that there's color coding. And if we detected the compound in the influent, we code it as gray. And if we detected it at a higher concentration in the effluent, then it's coded as red. If the concentration decreased during treatment, it's coded as blue. So what we really want to see in wastewater treatment is that all of these effluent concentrations should be blue, suggesting that concentrations are decreasing due to treatment. But what we see here is there's a lot of red, which suggests that we have slightly higher concentrations for some reason, and I'm going to talk about that a little bit, slightly higher concentrations in the effluent than we see in the influent. So one of the reasons we might see that is because our influent samples are really dirty. It's a dirty, what we call a dirty matrix. It's really hard to analyze for really low-level compounds like PFAS in dirty samples. So we don't have as good of a measure in the influent as the effluent, which was much cleaner. We also have a lot of biological processing going on in the facility that can liberate some of these compounds from solids. And that might also be one of the reasons that we see the difference in the influent and the effluent. So I'm going to talk about that a little bit more. When we looked a little bit deeper in the unit process, and this is where we actually look within the facility and say, how are these compounds changing in terms of total concentration as they move through the facility? So in July, when we did this more extensive study, what we noticed was that before the biological treatment or in the influent of these facilities, we detected the lowest concentration. And then after the biological process, secondary treatment, concentrations increased in all of the facilities that we looked at. And this is suggestive that there's something happening in that biological treatment basin that's again liberating those compounds or partially degrading some of the larger compounds to smaller compounds that we might be measuring. So we saw a dramatic increase in the total sum of the PFAS that we were measuring in all 4 of these facilities in the water phase, the phase that will be discharged to surface waters after this biological treatment piece of our facilities. We also see that the primary type of PFAS that we're detecting are these light green colors, which are these perfluorocarboxylic acids. So, and generally they're smaller, which means that they represent, they have a smaller backbone piece than some of the other compounds that we were looking for. So they're smaller compounds, they're primarily PFCAs that are part of this aqueous piece of our wastewater treatment facility. And I mentioned that we applied this new technique called a top assay that helps us better close the mass balance. So what we wanted to know is if we measured our samples using the top assay, did we detect more mass than measuring it using the isotope dilution method where we're not pre-oxidizing that sample? And the answer to that question is yes, we detect a lot more mass in these samples when we pre-oxidize it using this oxidizable precursor assay. And what this means is that there's a lot more mass there that we're not seeing using our targeted approach. We don't necessarily know what those compounds are. That's potentially transformable during the treatment phase. So for example, if we look at the total influent concentration in March, we see that there's almost always higher influent concentrations in the black bars. And it really didn't matter which compound that we were measuring, except for PFOA, one of these really terminal breakdown products. So we're always seeing more mass in the influent, usually, than we are the effluent, if we use this oxidizable precursor method to oxidize that mass to get a better indication of what's there. In July, we don't have as much oxidizable mass, and we're attributing that to the fact that there's more biological breakdown in the system in the upper sewer shed due to warmer temperatures and biological activity before it reaches the facility. If we're to sum that up over on the right-hand side, what we see is that we have about 6 times more mass. We see 6 times more mass using our oxidizable precursor method than we do using our targeted approach in the influent. Much of that goes away in the effluent, primarily because there's biological oxidation occurring in the treatment facility, so we reduce that oxidizable mass before it's discharged. And then again in July, we see less oxidizable mass, primarily because we think it's already being oxidized upstream in the system before it's discharged. And you can see the surface water does have some oxidizable mass in it, but it's lower generally than the effluent during certain times of the year. So I know that's a lot to think about. The other piece of this is, really understanding the difference in the PFAS composition in the aqueous phases, which are influent and effluent, versus the solids phases. And so I mentioned that we took samples of the sludges, and one of the fascinating things about the composition of the sludge is that it differs dramatically in terms of PFAS composition and concentration than the aqueous phase, which is the effluent. And so what we see is in the influent, we have a mixture of our perfluoroalkyl acids and precursors. But in the effluent, in the aqueous effluent, we have a lot of shorter chain compounds that are dominated by these carboxylic acids. Whereas the sludges, we see many of the longer chain compounds, sulfonic acids and precursors. And so what this tells us is that as these compounds are coming into the facility, they're being what we call fractionated. So the shorter chain compounds and the breakdown product, some of those terminal products are being discharged in the effluent. And the other longer chain compounds, some of those precursors and the sulfonic acids, those are being fractionated and going to the sludges, which are then going out a different way for the facility. And the partitioning is related by something that we call the log KD, which is the absorption coefficient that tells us how much these compounds like solid versus liquid phases. And we saw these really big differences in the aqueous phase versus the sludge phase across all the facilities that we looked at. So if we kind of sum up all the short chain versus the long chain compound, we can really see that as the PFAS move through our facilities, they come in a general composition after biological degradation. We have more short chain compound in the effluent, and that short chain composition grows until they're discharged, whereas the sludges are primarily dominated by those long chain, those precursors, and those fluorotelomere compounds. And then finally, a really important piece of this is that as we're fractionating these compounds into the sludge, what we see is that they're accumulating in the sludge at concentrations considerably higher than are being discharged in the effluent. And so we generally see concentrations about 1,000 or more fold higher in the sludge than we do in the aqueous phase. And this is important because even though the aqueous concentrations are much lower, we're discharging a much higher volume of those on any given day than we are the sludges. And so in general, this is actually a good thing to be able to concentrate contaminants in a solid form for handling, as long as that sludge or solid is handled appropriately. All right, and so if I'm summarizing what we've found so far, and we have a lot of other research ongoing, what we see in these facilities is that PFAS seems to fractionate during treatment. We have dissimilar signatures in the effluent and the sludges of these compounds. The effluent's dominated by short chain, more mobile compounds that are carboxylic acids, whereas the longer chains and the precursors are going into the sludge solid form, and the sludges are much more concentrated in the aqueous phase, although we do generate much lower volumes of those going out of the facility. So I just wanted to close by saying that, these facilities are really critical for collecting our wastewater from many different sources that contain PFAS, and then processing them in a way to reduce environmental contamination from these compounds. But we see that there's a lot we don't know in terms of what's happening in the facility. We know that the treatment process and the sludge handling is probably playing a really important role in what's going out the pipe in terms of discharge, as well as where and how much of that is concentrating in the solids form and where those solids go in the environment. And so source reduction upstream is going to be really important to reducing the mass that comes into the facility and is then hopefully reducing the discharge in the solids and the liquid form. And there's a lot of research that's needed to understand how these compounds are transforming, what compounds they're making in these facilities, and also how our facility design and engineering influences the fate of these compounds, both in the facility and outside of the facility. And we also need to do a better job of accounting for the mass coming in. Right now, we're looking at a very small number of compounds, and we know there are many more that are likely part of our wastewater. So, so I think in terms of time, I just wanted to stop there and thank my funding agencies, the New Hampshire Sea Grant, as well as the partners and students that are involved in this research. And I will pass it back to John so that we leave time for everybody to give their talks. Thanks.
Great. Thank you, Paula. And now I'm going to make Celia a presenter.
Okay, second.
Yep, we can see your screen. Yep.
Oh, good. Okay, I am going to move hold on one second, our pictures down so I can see my screen too. Yeah, well, that was great, Paula. I am taking notes rapidly because I learned a lot from that. And I guess I'm going to take you to the next stage of where the effluent ends up going and tell you about the research that we're starting. We're relatively new in doing research on PFAS. I'm an aquatic ecologist at Dartmouth in the Department of Biological Sciences. And I've been studying contaminants in food webs for quite a while, mostly mercury over the last several decades. But today I'm going to tell you about our new research projects on PFAS and share with you some of our preliminary data from Great Bay and from the Gulf of Maine. Since these are unpublished data and they are preliminary, I'm going to show you, parts of data sets. I would appreciate it if you wouldn't distribute this to any of these data to others. You can always contact me about that. So let's see. I'm just going to show you a timeline because you can see from this timeline how short a time we have actually been working on PFAS. We started in September 2020, and my colleague who's here with us, Dr. Megan Romano, and I and two colleagues at Clarkson received an NIH grant that we allowed us to start to look at PFAS in Great Bay. We later were able to get money to bring in a technician, which was enormously helpful. And we just in the last several months received New Hampshire Sea Grant funding and funding from the DOE and Department of Energy, Department of Defense Strategic Environmental Research and Development Program. As you can see, similar names as collaborators, and Dr. John Patali, who is our host here, has been a collaborator with us throughout. We also just received a grant to look at PFAS in freshwater ecosystems. And I should say that it's been especially interesting for me because a lot of the questions that we are posing in these grants are parallel to the ones that we asked about mercury. And so of course, we know a lot more about mercury, but now we are learning whether the processes in these food webs are similar at all. So first, I'm going to just take you to the first grant, which was the NIH grant, and show you where we started. And here we were looking at three things. We were looking at a sample set of fish that we had collected from the Gulf of Maine for another grant. And looked at PFAS in those fish. I'll show you some data momentarily. We're also looking at the concentrations in bivalves near and farther from the former Pease Air Force Base. And then we also conducted a Gulf of Maine, a seafood survey, which Dr. Megan Romano will tell you more about later. So the samples that were collected in the Gulf of Maine, here you can see the Gulf of Maine, we collected what we are calling inshore samples of the different species that you see up in the legend up here: silver hake, red hake, spiny dogfish, skate, herring, and Atlantic cod. But we also were able to, and I shouldn't say we collected them, we were able to get fish from the National Marine Fisheries Service during their demersal fish surveys, and also worked with a fisherman in New Hampshire, David Gaithel, to get some of our inshore samples. And so we had a sample set of these different kinds of fish, and we had been studying the metal concentrations in them, but we were then able to freeze them and now use them for Pfas analysis. The next slide shows you our preliminary results for these fish. And what you can see from this is that these are data for only 44 out of the 153 fish that we have. And what you can see is on the graph on the x-axis are the fish species across the bottom. And then we also have then for each of the compounds, PFNA, PFOA, PFHXS, PFBS, and Pfos, these are all different compounds with different chain lengths, as John and Paula were talking about. You can see that what we have are the dotted lines are the EPA screening levels, the green dotted lines, and we have the blue dotted lines, which are the New Hampshire finfish screening levels. And what you can see from this is that the Pfos concentrations, which are the blue bars, for all the inshore fish are actually higher than the New Hampshire screening levels. And in the case of, so the solid, I should say, the solid are the inshore, the bars, and the hashed are the offshore. So really the inshore we see, and we see this with metals as well, that the inshore fish, which are the same species as the offshore, have higher concentrations than the offshore, which kind of makes sense if it's really from coastal sources of Pfas. Then if we take a look at the sites where we collected shellfish, and here you can see we have dots representing where AMAC Foster Wheeler had collected for the former Pease Air Force Base, had collected bivalves, and then we also have the sites where we collected bivalves. And really, we could never have done this without the help of our collaborator, Chris Nash, who's at the New Hampshire DES. We barely really even knew how to collect shellfish. We had not worked with shellfish in the past. And so from there, we actually then collected from these different sites. And you can see in the red circle is where the former Pease Air Force Base is. And so areas like Woodman Point and some of these other sites, Little Bay, Fox Point, are in the watershed of the Pease Air Force Base. So when we looked at the data for actually the sediment and water that were collected by AMAC Foster Wheeler, you'll see in this next graph, we compared them to the sediments that were found at an AFFF site, the firefighting foam site in Canada, another study that was done in 2017, and the water concentrations in those sites. And you can see as percentages of the measured PFAAs that the sediments had largely Pfos and smaller percentages of the other compounds, but there was a mix of the different compounds in water. And here is the kind of breakdown of the compounds from the Pease Air Force Base samples that were taken by AMAC Foster Wheeler. And you can see here that in the sediments, it's all Pfos, and in the water column, you also have this mix of different compounds, but dominated by Pfos in the water and PFHXS. And so there is some similarity between these abiotic compartments in terms of the compound profiles. The reason that this is important to us is because in the estuary, we are studying animals that eat from the sediments and from the water, and we're interested in knowing if the sediment compounds are reflected in the tissue concentrations in those organisms that eat in the sediments, and if the water compounds, concentrations and compounds are reflected in the organisms that consume food out of the water column. If we look at the data then for the compounds, now this is way too much information, and I don't really want you to look at all the different compounds. I just want you to notice that in fact here we have blue mussel and oysters, which are both species that feed relatively in the water column. And then we have sediment burrowers like the razor clam and the soft shell clam on the right that are really feeding in the sediments. And what you can see in all four of these circles is that the compound breakdown is extremely different between each of these different kinds of shellfish. And in fact, there isn't any more similarity between the blue mussels and the oysters that feed relatively from the water column as compared to the razor clams and the soft shell clams. The other thing that you'll see, I've put the concentration ranges. The concentration ranges are different. There's much bigger range in the blue mussels and in the soft shell clams, and less so in the oysters and the razor clams. But one thing that we find interesting, relative to what Paula was saying about the precursors, we see an enormous amount of precursors in terms of percentages in the soft shell clams. And so these are the kinds of things we're wondering about and trying to understand as to what the profile of compounds is, as well as what the concentration ranges are in these different organisms. And so I think we found now that compound profiles vary by shellfish species. They don't vary so much by feeding strategy. They vary more in blue mussels and soft shell clams, and that the precursors are pretty important in the makeup of these. If we look at the concentrations of the different compounds in these by site, here you have the sites on the bottom on the x-axis, and you can see the compounds then are the different colors. You can see that Woodman Point, which is this last set of bars on the right, is actually lower than some of the other sites like this LBFP1, which is up also in the watershed, but not even as close to the site as Woodman Point. And the others, like these LH8WM and LHOP2, which are really in a different part of the Great Bay ecosystem, actually have, medium amounts of Pfas in those bivalves as well. So the site differences don't seem to be necessarily related to distance from the Pease Air Force, former Pease Air Force Base. But as I'll show you here, for PFOA on the left and Pfos on the right, and we have the screening levels listed here. The screening levels for both New Hampshire and EPA for PFOA and Pfos are well above. They are, upwards at the top of the graph at the 1 level, and well above the concentrations that we're finding in the bivalves in those sites from Great Bay. And then the same is true for the PFHX as well as the PFBS. And so I guess at this point, we don't see very high risk concentrations in these different bivalves, except for in one site at Little Bay Fox Point 1, where the razor clams are really significantly greater than the concentrations for Pfos than the oysters or soft shell clams. So that's something interesting. We don't know why the razor clams are higher, but that's what we're finding so far. So now I'm just going to tell you about the CERTIP and New Hampshire Sea Grants. that we actually have just gotten funded and have literally last 2 weeks have been sampling. And what you can see from this map on the right is the sample sites. And we are actually, this is been a bit modified, but what we have in our sampling are several sites that are downstream of wastewater treatment plants, literally at the outfalls of the wastewater treatment plants, and we have sites that are within Great Bay. And here we want to know what the profiles and concentrations, like I just showed you for the bivalves, are for sediments and water, as well as other organisms in these estuarine food webs. We also want to look at the impact of organic carbon on the PFAS in the sediments, as well as whether it changes the amount that's taken up by organisms in the sediments. We saw that with mercury, methyl mercury, that when there was higher organic carbon in the sediments, that there was a lower bioaccumulation into the organisms associated with those sediments. And then finally, we are also proposing, and we'll do chronic and acute toxicity testing for several PFAS compounds with lower trophic level marine organisms, and look at the relationship to carbon. So I'm going to skip over one of my slides, but here is what we're looking at for abiotic and biotic PFAS concentrations. You can see some of the organisms that we have been collecting, including water and shellfish. We are collecting a lot of killifish. We've been out and found killifish in all of our sites. We have green crabs, blue mussels, rib mussels, periwinkles, mud snails, a lot of different organisms. We will also be measuring stable isotopes, because that gives us an idea of how high in the food chain each of these types of organisms is feeding, and also where their carbon source is. Is it mostly from the sediments, or is it from the water column? Our experiments that I described earlier, we will be getting sediments from one of the more contaminated areas in Great Bay and adding sand so that we have a range of organic carbon. Here you can see 1.6% up to 6.5%. It's measure of loss. It's called loss on ignition. It just means the 6.5% has higher carbon in it. And we will then be using two different organisms, an amphipod species called Leptochirus plumuliosus, and we in experiments to see how much they take up from each of these sediment conditions, as well as Fungulus or killifish, which we will also be testing in the lab. And then lastly, we will be looking at these chronic and acute toxicity tests using a microalgae, a marine microalgae, and a copepod species, as well as our trusty amphipod species, and looking at 4 different PFAS compounds, both looking at acute and chronic toxicity, and exposures. With the aqueous exposures will be, water exposures will be for the microalgae and the copepod, and the sediments we will use to expose the amphipod species. So hopefully I haven't taken too long, but here's a list of our funding sources. Here is Adams Point, which is just a gorgeous place to get to go and sample. Our funding sources from the NIH, from Nih Grant, from DOD and DOE, and from the New Hampshire Department of Environmental Services. And none of this work is done without lots of other colleagues and collaborators who are listed below. So thanks for your attention.
Great, thank you, Celia. And last but not least, I will go ahead and hand this over to Megan.
Did I get myself off of?
Yeah, you're exited out, and yep, Megan, your screen is up and looks good.
All set. Awesome. Thank you. Well, thanks everybody for being here this evening. And I'm going to tell you a little bit about some of the PFAS and human health research that we have going on in the state of New Hampshire. So I won't belabor this too much because John actually set it up very nicely in his introduction. But this is a figure from a recent open access review article that was led by Sue Fenton from NIEHS that describes really nicely what we know about the effects of PFAS on human health. So here in this graphic, the solid lines represent high certainty associations and the dashed lines are lower certainty associations, where we still need some more scientific studies to help us understand how PFAS may influence those health outcomes. And as you can see, there are a lot of important open scientific questions over on the left side where we see the woman and her developing child. And this is also where most of my research resides. So currently I'm engaged in two interrelated lines of research that are related to PFAS. The first has to do with understanding potentially underappreciated sources of PFAS exposure. And in this line of research, I work with Dr. Chen to understand the influence of seafood consumption on PFAS exposure. And I'm also actively working on describing PFAS in breast milk as a potential source of exposure to newborns and infants. In the second line of research, I use the New Hampshire birth cohort study to investigate the influence of PFAS on multiple facets of maternal and child health. And I'm really excited to be able to share a bit about some of these ongoing projects with you this evening. So first, thinking about seafood as a source of PFAS, which was so nicely introduced by Dr. Chen in the previous presentation. We recently used an adaptation of the Idaho Fish Consumption Survey, which was created in accordance with US EPA guidance on the conduct of reliable seafood consumption surveys, geared towards a purpose of establishing surface water quality standards. And we used that survey to help us understand fish consumption patterns among New Hampshire residents. For this work, we leveraged the Granite State Panel, which is reflective of the adult population of New Hampshire. And members of the panel are recruited by the University of New Hampshire Survey Center from randomly selected landline and cell phone numbers across New Hampshire. Panel members are also recruited by texting a random sample of cellular telephones in the state and inviting those recipients to take a short survey online. Our online survey asks questions about sociodemographic factors, frequency, types, and typical portions of seafood consumed. And the respondents provided information about their own seafood consumption, as well as seafood consumption by children aged 2 to 11 years old that resided in the surveyed households. We launched the survey on June 3rd of 2021 and had 1,829 respondents that participated. And I will give you just a tiny preview of our preliminary findings from this work. So perhaps unsurprisingly, given that we are in the northeastern region of the United States, we found that fish and seafood consumption were common among both the adult respondents and the children between the ages of 2 and 11 years that resided in those households. We also noted that a majority of the respondents indicated that their fish consumption patterns were not impacted by the COVID-19 pandemic. We were really interested in asking this question because we're aware that a lot of people have kind of changed their behaviors and food habits. A lot more people are cooking at home. A lot more people are also getting takeout and sort of eating differently. So we wanted to just sort of make sure that this was representative of overall seafood consumption, not strictly seafood consumption during the pandemic. And it seems like that is the case. We found that among people reporting regular consumption of seafood, haddock, canned tuna, scallops, sushi, and clams were among the most common types of seafood consumed. And on this graph here, it's just showing you sort of the percentages of different people reporting different types of seafood that they were consuming in the time leading up to the survey. So what we're planning to do is to integrate this survey information with data from a market basket experiment that will measure PFAS in these commonly consumed types of seafood that we've identified. We have purchased a number of samples from local fish outlets and seafood providers, and we're currently working on doing those Pfas measurements in our laboratory. Once we have that data, we'll combine it with what we learned from the survey to help us better understand how fish and seafood consumption may influence Pfas exposure among New Hampshire residents. All right, so next I'll tell you a little bit about our work studying Pfas in breast milk. So in 2018, we received a Human Health Exposure Analysis Resource Award that allowed us to quantify Pfas in human milk samples from the New Hampshire birth cohort study. I'll tell you a little bit more about that study a little later in the presentation. Through this award, we were also able to generate untargeted metabolomics data so that we can further investigate the composition of human milk. We're particularly interested here in understanding the correlation between Pfas measured in blood during pregnancy and Pfas in human milk following pregnancy. And so these graphs come from a manuscript that we're just getting ready to submit. So on the y-axis, we have either PFOA in the breast milk sample or Pfas in the breast milk sample. And then on the x-axis, we have PFOA in maternal plasma that was collected during late second trimester in pregnancy, or Pfas plasma on the x-axis. And so what we note here is that there is a very strong correlation between the level of PFOA and Pfas that we find in the breast milk according to what was present in mom's blood. Quite strong correlations, 0.8 for PFOA and 0.77 for Pfas. But what was very interesting was that when we look at the absolute concentrations in the two different matrices, we see that the breast milk had about 2% of the PFOA versus maternal plasma. And for Pfas, the breast milk had only about 1% of Pfas versus maternal plasma. We did look at a variety of other Pfas, and the majority of them were not detectable in our milk samples. And so even though we're seeing that not very much Pfas is making it into the human milk as compared to mom's plasma levels, given the high volume of fluid that infants take in per body weight, this may still be levels that we need to be carefully considering when we're thinking about health effects, particularly in those newborns and infants. There aren't clearly established guidelines or thresholds for Pfas in terms of where we think about those levels being in human milk. So again, while it's good news that there's comparatively little Pfas getting into the milk versus the maternal blood, this small amount of Pfas may still be important. And so we are planning some additional studies to sort of continue unpacking that relationship between Pfas and human milk and infant health. And that includes an upcoming conference presentation at the International Society of Environmental Epidemiology. Their annual meeting is in Athens, Greece this year. And Hannah Lowy, who is a postdoc in our department, will be presenting her work exploring early life exposure to Pfas from breast milk and gut microbial composition at that meeting. So we're really looking forward to seeing that research come out and continuing to think about how these levels of Pfas in milk may influence child health. So more broadly, thinking about the work that we're doing, looking at Pfas in the New Hampshire birth cohort study. Just a little bit of quick background on the study itself. The cohort PI is Dr. Margaret Karagas, who's our department chair in the Department of Epidemiology, and she started this study in 2009. And so since 2009, the New Hampshire birth cohort study has been investigating how the influence of environmental contaminants might affect the health of pregnant women and their children. So women are recruited from clinics in the Lebanon and Concord areas of New Hampshire during early pregnancy. We will also be adding a recruitment center in Manchester in the next couple of years. And these women are going to be followed through their pregnancy. The children are presently followed through about 12 years of age, and we have plans to extend the follow-up into adolescence over the course of the next 2 years. We'll be rolling out that adolescent life stage. So broadly, we investigate how environmental factors influence complications of pregnancy, birth outcomes, physical growth, neurodevelopment, and many other aspects of maternal and child health in this study. And so in 2016, I was able to launch a project specifically looking at Pfas in the New Hampshire birth cohort study. Now, this project was funded through a Center of Biomedical Research Excellence grant, which funds the Center for Molecular Epidemiology at Dartmouth College. And here I was interested in some interrelated aims, understanding how maternal plasma Pfas in pregnancy influences maternal gestational weight gain, so mom's own weight gain during pregnancy, the influence of that exposure on breastfeeding duration, as well as breast milk composition, which you saw a little preview of some of that work, and also understanding how those exposures to Pfas prenatally influence adiposity in early life. So we're hoping to publish most of this work in the next 8 to 12 months. So definitely stay tuned for updates in those regards. To that end, some manuscripts that we have in the pipeline are one manuscript, oops, sorry about that. One manuscript being led by one of our wonderful undergrads looking at some NHANES data, trying to understand the association between fish oil supplements and Pfas in NHANES. We also have a manuscript submitted looking at the Pfas exposure and infant growth trajectories in the New Hampshire birth cohort study. Some additional things that we have in the pipeline will be our first paper looking at those fish consumption patterns among New Hampshire residents. So that's going to be primarily geared at understanding just those patterns of fish consumption, and later on we'll be publishing a paper integrating the Pfas data in to understand that specifically as a potential source of exposure in New Hampshire residents. We'll also have a paper forthcoming describing that maternal plasma and human milk correlations in the birth cohort study, looking at gestational weight gain, gut microbial composition, and then Kyun Yim, who is one of my postdoctoral fellows, is working on a project trying to understand how mixtures of heavy metals and Pfas influence birth outcomes in the New Hampshire birth cohort study. And so she'll also be presenting that research at the ISEE conference that's upcoming in September, and we're anticipating that the manuscript will not be too far behind. In addition to that, we have been working on some funding proposals to study human milk specifically in Pfas contaminated communities, as well as being able to look at a wider range of cardiometabolic health outcomes in the moms postnatally. Those are pending, they're not funded projects yet, but we're really hoping that that's work that we're going to be able to do over the course of the next 5 to 10 years. And so with that, I will just say a quick thank you to my many wonderful collaborators. Like Celia, it takes a village. I'm sure I haven't said thank you to all of the wonderful people who have contributed to this work, and acknowledge my funding sources. So I guess with that, I'll turn it back over to you, John.
Great, thank you, Megan. So with that, we'll go ahead and roll right into the Q&A session. So if I can call Paula back up. I tried to say Paula Mauser too fast at the same moment. It came out as one word. So it looks like we have 2 questions in the chat already, and I'm going to pop these out, and I'm going to scroll up to this one. Sorry, I'm going to lean in to make sure I read it right. And this first one looks like it's for Dr. Mauser. What can communities do to reduce the amount of Pfas in effluent water to stop further contamination back into the environment? And what do the wastewater treatment plant systems need for additional treatment systems? And where should the solids go after contamination? There's a few other questions after that, but I think I'll let you answer those 3 first.
Great, thanks so much for these questions. Yeah, the first question I think is really important because in the past, one of the ways that we've combated environmental contaminants is looking at the sources and reducing the sources so that the sources don't contribute to the spread of the contamination anymore. And so as communities, one of the things that you can do is you can look at possible funding through the New Hampshire DES and the Wastewater Bureau. There are funding sources available related to characterization of Pfas and in the upper sewer shed in your municipal wastewater treatment facilities to characterize where you might be having sources coming into your wastewater system, and then and working with your town public works folks to try to reduce some of those sources. And I'd be happy to point you in the right direction for who you might want to contact or assist with helping with those characterization studies. We actually are doing this with the Vermont DEC right now. We've characterized Middlebury as well as Essex Junctions, and we're seeing really interesting research related to the fact that a lot of the sources are coming from residential areas in the sewer shed, and that's because it's fairly easy to get rid of that low-hanging fruit. Some of those industrial sources might have already been targeted and reduced. But now we have a lot of these kind of very dilute sources that we're using in our households that are contributing in the sewer shed that are a little bit more difficult for us to remove. And so I think these upper sewer shed characterization, source characterization studies are really important for communities to identify the low-hanging fruit and reduce at the source PFAS coming into their facilities. So the second question related to treatment, and this is also a really important part of regulatory kind of policy ongoing right now at the federal level and the state level. If it is decided to regulate PFAS in source waters, what that means is that the wastewater treatment facilities are going to have to monitor and meet regulations that are tied to their discharge permit. Some of them might be required to install additional treatment, and generally in a lot of our treatment facilities, that treatment might be something like granular activated carbon, which is a piece that we might add on to the end of the treatment facility to try to capture some of the very small compounds in the that might be coming out in the effluent. So there are some options. They're not foolproof, and it's very difficult because the treatment options don't necessarily target all of the compounds that we're looking at in these facilities. So I think I hit the first couple, John. Was there another piece that I didn't get to?
So, and then the other piece was, and where should the solids go to prevent further contamination? How can we make sure that solids are not used for agricultural purposes? Sort of, she has a question mark in there asking regulations question mark, and
Right.
further contamination of food sources, animals, soil, etc. Andrea, if I missed anything, just shoot me a message in the chat and tell me what I missed.
Mm-hmm. That's such a great and also ongoing question in the regulatory realm right now. A lot of states are looking at regulating PFAS in solids, biosolids specifically. Maine has set some limits that are very low. Massachusetts is going through a process through modeling to try to establish these limits. I think that some of this is ongoing at New Hampshire DES. Maybe John can comment on that. Ultimately, the process for basically collecting contaminants in solids and then handling them appropriately is actually a really good thing. And it's not just PFAS that are being collected in these sludges. There are other compounds, and they are processed in a way that allow further reduction of pathogens and contaminants. And so there's a huge amount of research that can be done that is ongoing, I think, in biosolids and biosolids processing to characterize what's there and to look at what processing methods might further reduce these compounds before release to the environment. We have a paper that came out last year that looked at PFAS compounds and biosolids that were processed different ways. So I'm happy to share those resources with this group as well. But this is a very active, ongoing research area. And so there are some treatment methods on the biosolids processing side that could also further reduce PFAS contaminants being released back to the environment through, say, biosolids application.
Sure, and thank you for that. And I know that we do have a program that is looking into biosolids. I do not know the granular details of what they are doing, so I don't want to misquote their work. But Andrea, if you want to get in touch with me after this, I can connect you with Anthony Droon, who's running that. Or maybe that'd be something we could do another one of these webinars in a few weeks, and we can bring Anthony on, and he will take up 30 minutes easily telling us about the work that DES is doing with biosolids right now. So I'm going to go on to the next question. So this one is either for Dr. Chen or for myself, and it's asking, when thinking about relating eating shellfish that were below the EPA or the New Hampshire screening levels, do we factor in communities that already have exposure through contaminated drinking water? So I'll go and take the first part of this question. The answer is yes, we consider other sources of exposure. With the screening levels that we develop, those are about 10 times lower than what we would even use to trigger action. So when we talk about screening levels, an analogy I like to use is think of a traffic light. So a red light would be a consumption advisory or a restriction we would put in place. Our screening levels work like a yellow light. It's telling us to look into this more. So even when we look at the margin of safety that's there at the yellow light for our screening levels that New Hampshire currently has, we still wouldn't be worried, but it is telling us we do want to look at this some more and just try to really understand what's going on. And that's why we've been partnering with Celia and Megan, is to really dig into this and understand not only what's in the shellfish, but also to look at what do fish and shellfish consumption patterns actually look like in our residents. Because a lot of times when we develop this guidance, we're relying on national data because historically New Hampshire hasn't had local data. So that's one of the great things of this recent partnership and this data that Megan is working on, and we're really looking forward to using this, is it's going to give us a better understanding of what our risk scenarios look like here, and we'll be able to refine those better. So in the second part of this question, you're also asking about, given the recent lowering of the lifetime health advisories for PFOS and PFOA, can the community expect the screening numbers for shellfish to go down from the EPA and or NHDS? And at this time, we don't know, because EPA has made very clear that, one, they are going to change that health advisory in the coming months, but they've also told us that they're not using that health advisory as the basis for driving fish consumption guidance or other guidance at this time. Now that may change in a few months, and we're looking to see what they do, but as they're going through their review process for those advisories, they're going to be changing those numbers, and we're looking to them to see what they're going to be changing around that. So in the interim, we're retaining our lower screening levels that are lower than the historic EPA ones. They're similar to the current, this is getting a little in the weeds, but they're current ones that they're using for the Pease Air Force Base. We see similar results when we look there. But at this time, we won't know until EPA finalizes their assessment of PFOA and PFOS in the fall for those screening levels. And then the next question is for Dr. Romano. Have you published the data on PFAS levels and common sources of fish products?
No, not yet. So we expect that to probably be out in the next 6 months. And if you are interested and want to email me and ask me to give you a first look once it's published, I'm happy to do that. For any of the stuff that's coming out, if people are interested in it, feel free to shoot me an email and I will make sure you get a copy when it's published.
Okay, great. And what I'll do is there's going to be an automatic email that comes out at the end of this presentation, or it'll be a couple hours later. I'll include everyone's contact information in there so you can follow up with them. Looks like the next one is from Patrick Carroll from Representative Papp's office. No question, just wanted to say thank you for all the hard work you all have done here. It means a lot to us. And the next question is anyone in New Hampshire studying PFAS in rainwater? How about dust in households, offices, and schools? Another question for Dr. Romano. Do we know how much PFAS passes from the mom to the baby in breast milk? Sorry, I bled those two questions together. So let's start with the first one on in rainwater. Did someone want to take that one?
Well, I can just say that some folks at a wastewater treatment facility reached out to me asking about sampling for PFAS in rainwater at the facility. And so I think some of the communities are starting to look at some of these other sources. But other than that, I'm not aware of any data on PFAS in rainfall. I'll pass the buck to others if they do know about that in New Hampshire.
So I will say that the New Hampshire Department of Environmental Services, we have done some rainwater sampling. We're still looking at some of that data, and that's something that our Air Resources Division is looking into, or Kathy Beam specifically would have more information about that. So we're still looking at the data with that. I think we're still doing some additional data collection to verify what we're seeing there. As far as PFAS in dust in households, offices, and schools, I'm unaware of any testing that the state has been doing. I don't know if, Megan, if you have any sort of exposure work that anyone's doing around that in New Hampshire, or maybe colleagues in other states that are doing any kinds of.
Not in New Hampshire, but I know Heather Stapleton's group at Duke has done some work with house dust, as well as Kate Hoffman's group. So more the North Carolina folks have been doing some really great work on house dust and hand wipes and kids too, which is somewhat a measure of dust. I'm not aware of anybody that's doing that research specifically in our state, but there is some really great research coming out of North Carolina that I think is probably relevant, even though we're not directly testing here.
Okay. And then the next one is for you, Megan. Do we know how much PFAS passes from mom to baby in breast milk? Can we predict, based on a mom's plasma level, how much PFAS will end up in baby?
Yeah, so this is kind of what we're working towards, to try and be able to understand, because ideally we'd be able to measure mom's PFAS during pregnancy and then get a sense of what that breast milk exposure might look like for the kids. We are doing some sort of back of the envelope type calculations where we're doing an estimated daily intake, where we have to make kind of a lot of assumptions about exactly what volume of breast milk kids are consuming in those early days. And there are some sort of standard assumptions that we can make based on FDA and EPA papers that have been published in this space, as well as some things that are coming out of EFSA in Europe. And so we do have ways to sort of estimate that and to try and understand it. But again, one of the other things that we really are trying to understand is, what really is the consequence of that exposure too, because there's a lot of really good stuff in breast milk also. And so we don't want to kind of lose sight of the fact that there are a lot of health protective and bioactive compounds that are being exchanged through mom and baby through that process of breastfeeding. And so I do think it's really important for us to kind of continue this line of research where we're trying to understand what those actual exposures from breast milk mean for infant health. And if that looks the same as when we look at sort of prenatal exposure based on mom's levels in pregnancy. And I think that that'll help us to understand a little bit more this question about how important is that PFAS in breast milk, even though a small quantity is transferring from mom into the breast milk, it's still important for us to understand the toxicity there.
Great, thank you, Megan. So with that, I'm going to check and make sure we don't have any more questions. I don't see any coming in. I'll give it just a few more minutes in case anyone else thinks of a last chance to stump the teacher. So again, I just want to say thank you to everyone for coming out tonight. So while anyone's thinking about any questions, there will be a follow-up email that'll come out after this. It'll contain contact information for the presenters here. If you have questions for anyone, I recommend just sort of reach out to whoever you think is right. We'll probably send you to the right person if you get the wrong one. There's no wrong answer. Just reach out to us. We're happy to help. This will also be, it was recorded, so we will try to get this online to either the DES website, DES YouTube page in the next couple of days. So if you have colleagues, friends, or anyone that missed this because of another obligation, or because they wanted to be outside on a really nice summer night in New Hampshire that wasn't in the triple digits, that will be available to them. So let me see if we have any more questions. Okay, I don't see any. So with that, I just want to say thank you to all the presenters for taking the time to come in tonight and share with us. And with that, I'll let everyone go and everyone have a good evening.
Thank you.
Take care.
Thank you, John.